Hello, my name is Dr. Boku. I'm a clinical geneticist at Children's National Medical Center in Washington, D.C. During today's presentation, I will discuss the genetics of Noonan syndrome with the hope of clarifying those complex genetic aspects of the disorder for patients and for families. My contact information is on this page for any questions that you may have at the end of this presentation. First off, as I'm sure you know very well, Noonan syndrome is a relatively common genetic syndrome, and it affects approximately 1 to 2,000 or 1 to 2,500 newborn babies. It is the second most common genetic cause of congenital heart disease, only after Down syndrome. It is a developmental condition because it affects the development of many body organs. Many of the common symptoms encountered in individuals with Noonan syndrome include, by far, more commonly, heart defects, most common of which is pulmonary valve stenosis with abnormally formed valves, as well as hypertrophic cardiomyopathy. Some individuals exhibit poor growth, especially in the first 1 to 2 years of life, and some can have short stature. Many individuals face some developmental or learning challenges and need individualized therapies. Just a brief overview of the inheritance of Noonan syndrome in general terms. Noonan syndrome is inherited as what we call an autosomal dominant condition. I'm just going to overview very briefly the basics of chromosomes and genes for everybody to be on the same page. Every cell of our body contains 46 chromosomes, which are arranged in 23 pairs. Each pair of chromosomes is composed of one chromosome from our mother and another from our father. Every chromosome has specific areas on it, which are called genes, which have specific functions in the body. Each gene has a copy of it. So we inherit one gene from our mother and the other copy of that gene from our father. In an autosomal dominant condition, only one abnormal copy of a specific gene is enough to cause the condition. This is the case for Noonan syndrome. One individual affected with Noonan syndrome has one abnormal copy of the particular gene and one normal copy in every cell of their body. What we know of Noonan syndrome is that approximately 50% of the cases are inherited from a parent who has the disorder. The other half happen new in an individual, which we call de novo. This means that this individual is the first one in their family to have Noonan syndrome. This is a random event that happens during fertilization. The recurrence risk, meaning the risk of having an affected child for every individual with Noonan syndrome, is 50%. This schematic shows the risk, which is random, with every pregnancy. This shows that one of the two parents has Noonan syndrome and the other one does not. With every pregnancy, there are 4 possible combinations of those chromosomes or genes. If we take into consideration every possible combination of those genes, about half of the times, this couple will have a child with Noonan syndrome, and the other half of the times the child will not have Noonan syndrome. This is again a random event that happens by chance with every pregnancy. Noonan syndrome, as you very well know, is a clinically variable and very genetically heterogeneous disorder. This means that not all of the clinical features are present in all of the affected individuals, and also that many different gene changes can cause Noonan syndrome. even with the identification of many of the responsible genes for Noonan syndrome, the diagnosis still remains clinical, meaning that it is based on identifying those key clinical features in the patients. The clinical diagnosis of Noonan syndrome can be confirmed in up to 75% of the cases, with finding a change in one of the corresponding genes, period. As a result, we know that approximately 25% of cases, or 1 in 4, do have Noonan syndrome by clinical features, but we are not able to find a change in the corresponding genes. I'm sorry. As of 2014, 9 genes have been linked to Noonan syndrome. Specific changes in the normal lettering of those genes in every cell of the body that someone is born with cause the protein that is made by this gene to function in an abnormal way. Each and every one of those proteins play an important role in a biochemical pathway which is in action in every cell of our body, and that's called the RAS MAPK pathway. The genes responsible for Noonan syndrome include the lettering that is important to ultimately produce a protein which functions as part of this pathway. This pathway, which I'm sure you have heard a lot about, is very complex and involves many different players. It plays an important role in the development of many different organs of our body during fetal life, in the formation and also the establishment of interconnections between nerve cells of the nervous system, and it affects the overall function of various factors and hormones, which eventually help each cell of our body and groups of cells to grow. Here you see a very simplified schematic of the RAS MAPK pathway, showing some select protein players without creating a lot of confusion, because this can become very complex. I will quickly review how this all works normally. All the players' proteins are located inside each cell, as you can see right here. Every cell is separated from the outside by a thin membrane, which is called the plasma membrane, which is depicted in the light blue right here. On the plasma membrane, there are many different proteins, which are called receptor proteins, such as these on the left and further to the right. Those receptor proteins are important because they interact with outside factors, including growth factors and hormones, and that's where the signal starts. Once a growth factor binds to specific protein receptors on the plasma membrane of the cell, which is right here, then this triggers a series of actions after those proteins, as specific molecules are transferred from one protein to the next in line. The RAS proteins are depicted right here in the middle, which are important players in this pathway. They help transfer this biochemical signal to other proteins, such as the MEK proteins and the ERK proteins. Eventually, the signal affects the reading of important genes inside the cell nucleus, which contains our DNA. In this way, specific genes are turned on inside the nucleus, which then affect the formation and development of specific cells and groups of cells and eventual formation of organs and development in specific organs. These are the 9 genes currently known to cause Noonan syndrome. I will discuss briefly what we know about each one of them in terms of their clinical findings. Namely, those genes include the PTPN11 gene, the SOS1 gene, the RAF1 gene, the RIT1 gene, KRAS, BRAF gene, SIBL gene, the NRAS gene, and one specific change in the SHOC2 gene. The PTPN11 gene is an important player in Noonan syndrome. It was the first gene to be identified to cause this condition back in 2001. About half of the cases are due to changes in this gene. 2 specific genetic changes shown right here. These are termed the N308D and the N308S protein or amino acid change in the protein have been shown to cause either little or no intellectual disability in the affected Noonan syndrome patients. Unique to PTPN11 gene changes are some specific findings. Individuals with PTPN11 mutations tend to have more of the short stature, pulmonary valve stenosis, bleeding disorders, as well as an increased risk of developing leukemia, mostly during infancy. They also are shown to have lower frequency of hypertrophic cardiomyopathy, which is signified as HCM, and also lower frequency of ASD, which is atrial septal defects, which is a communication between the upper 2 portions of the heart. Specific different mutations in this gene cause a closely related syndrome called Noonan syndrome with multiple lentigines, which was previously known as Leopard syndrome. Those again are specific gene mutations in PTPN11, and individuals with this syndrome have characteristic findings of Noonan syndrome, but additionally, they also tend to have a higher frequency of sensory neural hearing loss, birthmarks such as skin lentigines, which are black, brown, flat lesions on the skin, mostly on the upper body, as well as heart rhythm abnormalities or arrhythmias. SOS1 is the second most common gene affected in about 10% of cases of Noonan syndrome. Important unique characteristics in this group of Noonan syndrome patients include a lower frequency of short stature, less of the atrial septal defects, less frequency of intellectual disability and speech delays, but we have seen a higher frequency of specific skin and hair manifestations, like the ones that we see in cardiofaciocutaneous syndrome or CFC syndrome. Those include sparse or thin curly hair, as well as some skin pigmentation abnormalities and thickness of the skin. Some patients can have the development of some benign lesions in the jaw bones or the mandible, which are called giant cell lesions. About another 10% of cases of Noonan syndrome are linked to genetic changes in the RAF1 gene. Unique features in this category include a higher risk of development of hypertrophic cardiomyopathy or HCM. Interestingly, up to about 2/3 of patients with RAF1 gene mutations end up having hypertrophic cardiomyopathy, and this usually develops in infancy or early childhood. This is in contrast to the general frequency of hypertrophic cardiomyopathy seen in all patients with Noonan syndrome, which is approximately 18 to 20%. Patients with RAF1 gene mutations tend to show more of the skin manifestations, such as birthmarks, including café au lait spots, which are brown even skin lesions. Some have many nevi or moles on the skin, and some can also have lentigines, those black-brown flat lesions that we can see also in Noonan syndrome with multiple lentiginous syndrome. Again, there is an overlap between Noonan syndrome and the Noonan syndrome with multiple lentiginous because there are specific different genetic changes in the RAF1 gene that cause the clinical features of Noonan syndrome with multiple lentiginous. RIT1 is the most newly identified Noonan syndrome gene just recently in 2013. It is thought that up to about 10% of cases of Noonan syndrome may be due to RIT1 gene changes so far. Only about 32 patients have been reported so far in the literature, making any conclusions on the unique characteristics of this group of patients very difficult because of the small numbers. The published articles about RIT1 mutations suggest a potential higher frequency of hypertrophic cardiomyopathy and also pulmonary valve stenosis in this group of patients, and possibly a lower frequency of short stature and intellectual problems. However, this will need to be studied in more patients as this is really now in the process. Another gene causing Noonan syndrome is KRAS. Mutations in this gene account for approximately 2% of cases, and some unique characteristics have been shown again. There tends to be a higher risk or frequency of more severe intellectual and developmental challenges in those children, and many of them tend to manifest many of the skin findings seen in cardiofaciocutaneous syndrome. Again, the hair findings such as thin, curly, and sparse hair, or the skin findings such as this thick, eczematous skin. Some mutations in KRAS have been linked to causing CFC syndrome in and of itself, so there is significant clinical overlap. Moving on to the BRAF gene, this accounts for about less than 2% of cases of Noonan syndrome in itself. Unique clinical characteristics in those patients include mild to moderate cognitive challenges, as well as, again, some of the birthmarks or skin findings, such as the moles and lentigines. Individuals with BRAF mutations are sometimes diagnosed with either Noonan syndrome or cardiofaciocutaneous syndrome. And in fact, up to 2/3 of patients with CFC syndrome harbor a BRAF mutation. It has been suggested that there may be what we call an intermediate phenotype in those children who have clinical features who fit somewhere between Noonan syndrome and CFC syndrome as far as their clinical severity. And so the actual diagnosis sometimes may be challenged or changed with genetic testing. Again, the CBL gene or CBL gene was identified again recently and accounts for only about less than 1% of cases of Noonan syndrome. Those patients tend to show a characteristic facial profile which resembles Noonan syndrome, and that's why they call it a Noonan syndrome-like condition. They also tend to have some unique characteristics again, which includes an enlarged left atrium, which is the upper left portion of the heart. They also tend to have more of the heart rhythm abnormalities, and maybe also a higher risk of developing leukemia, with the most common being JMML, or juvenile myelomonocytic leukemia. Mutations in the NRAS gene caused a small proportion of cases of Noonan syndrome, less than about 1%. Two specific gene mutations in that gene have been described, and they're depicted here. There are very few cases reported in the literature with NRAS mutations, so we do not have specific unique characteristics for those patients as of yet. Lastly, one specific change in the gene called SHOP2 gene, which is again involved in the RAS pathway, caused Noonan syndrome, but with some, again, unique characteristics. Those children tend to show specific hyperactive behaviors. They tend to have growth hormone deficiency in some cases and poor growth. They again have some hair and skin findings, but in this case, the hair are characterized as loose anagen hair, which means that they are thin and slow growing. And also the skin tends to have a characteristic hairless or dark pigmentation with some thickening, which we call ichthyosis. Those children tend to show a hoarse voice or a hypernasal voice when they speak. And also, in terms of their heart defects, they present more with dysplasia or abnormal formation of the mitral valve, which is on the left side of the heart, and more with those septal defects, which are communications between either the upper or lower portions of the heart in the form of an ASD of an atrial septal defect, or a VSD, which is a ventricular septal defect. As a summary, as you have seen, there are many different genes, mutations in which have been shown to underlie Noonan syndrome. It is important to note that there is great variability in the clinical findings of Noonan syndrome, even within the same family, in individuals who have the same genetic change. Then a negative genetic testing of all of the 9 genes that are currently available for Noonan syndrome does not rule out the diagnosis of Noonan syndrome, because it all picks up a mutation in 75% of the cases. As a result, the impression based on clinical findings becomes a very important. And finally, if a child has atypical findings for Noonan syndrome, it is important to also raise a suspicion or consider alternative diagnoses that may look like Noonan syndrome. Some other conditions that can look like Noonan syndrome include some of the ones I list here. One is what we call neurofibromatosis Noonan syndrome. This is a combined condition which is thought to be a subtype of neurofibromatosis type 1. Those patients have NF1 by the clinical criteria, meaning they will have the clinical manifestations of NF1, including café au lait macules, freckling in the armpits or in other non-sun exposed areas, and some of the complications of NF1, but they also have facial and physical characteristics like the ones seen in Noonan syndrome. Those patients, as far as we know, have genetic changes in the NF1 gene. Closely related to Noonan syndrome is Noonan syndrome with multiple antigenies, which sometimes can be difficult to diagnose in infancy and early childhood, as those litigenies tend to show up between 5 and 7 years of age. Other conditions that we have already mentioned include cardiofaciocutaneous syndrome as well as Costello syndrome, both of which are caused by changes in genes, again, of the RAS pathway. There is significant clinical overlap, and those sometimes are difficult to distinguish from Noonan syndrome, especially in milder cases. Now, outside of the RAS pathway, there are 2 specific syndromes that are worth consideration when someone is undergoing clinical genetic evaluation. One is Turner syndrome, which is exclusively seen in girls and is caused by the loss of one X chromosome. The girls with Turner syndrome have very similar facial and physical characteristics like Noonan syndrome, but they also have very different heart abnormalities and different other organ abnormalities. Lastly, Arskog syndrome is a rare genetic syndrome which can resemble Noonan syndrome in the physical characteristics, and it's also mostly seen in the boys. Thank you very much for your time. I hope that this clarified this complexity of the genes involved in Noonan syndrome. If you do have questions, I'm available at any time. You can contact me on my email, and I'll respond to you as soon as I can. I hope that this has been helpful for you. Thank you very much.